Welcome to a walkthrough of WEGAN's diversity tools for analyzing ecology data. WEGAN is a web-based platform for analyzing ecology data that requires no coding knowledge. In this step-by-step tutorial, we will show users how to use our diversity analysis tools, such as for performing alpha and beta diversity analysis. To get started, continue to wegan.ca.
To start, click on the Diversity module to explore diversity analysis methods such as rarefaction and abundance distribution. Next, upload your dataset using the Choose File button. Alternatively, users can use sample datasets. For this example, we will select the Aravo sample dataset. Selecting the Aravo dataset will also load the accompanying environmental data. Click Submit to proceed with your chosen data file. The Data Integrity Check page will confirm whether the data is usable, such as checking for correct sample and variable labeling. See our data uploading tutorial for more details on how to upload data and manage errors. Here we see the dataset has passed the integrity check. Scrolling down, the Data Overview page will also display a PCA plot and heat map of your data. Click the Heat Map tab to see a heat map overview, as seen here. The Data Editor tab can be used to make slight modifications to the abundance data. Here we can change the data type, edit the data, and remove entries. Find more details on our data uploading tutorial. Once ready to continue, scroll down and click Proceed. On the normalization page, you can select the normalization methods appropriate for your dataset.
There are 4 manual data transformation options to choose from. Click a radio button to select a methodology. Click Manual Normalization to apply your own data normalization configurations. A pop-up will show you variable data distribution plots comparing the effect of your selections. In this example, no normalization was applied. To make revisions to the normalization, click Go Back. Going back, we can alternatively choose Automatic Normalization to apply recommended normalization settings based on Pearson mode skewness. For example, here are the distribution results of the automatic normalization.
Click Export to save this plot. Select the export format from a drop-down menu. We can select from PNG, TIFF, PDF, SVG, and PostScript files. Next, select your desired export resolution. Options range from 72 dpi to 600 dpi. Finally, select export size of either full page or half page. Click Submit to generate the export file. The download link will now update. Click it to open the export file in a new tab.
To move on from the normalization step, click Proceed. For ease of replication, this tutorial will continue with no normalizations.
The second Data Integrity Check page verifies data quality in the supplementary data. This page is for our environmental data. Here we see our supplementary data has passed the integrity check. Click Proceed to continue. This third Data Integrity Check page is for our trait data. Again, our data has passed the check. Click Proceed to continue. After completing data pre-processing, you can select the type of diversity analysis to perform. This example will start with measures of biodiversity analysis. We have now reached the main analysis page. Here, we can choose our desired plot and customize plot parameters. We start with alpha diversity testing, which examines the diversity of a species within a specific habitat or ecosystem. Start by adjusting the variables and parameters of the plot. Select whether to input original data or normalized data.
Next, choose the data standardization method.
Finally, select a grouping variable for sample stratification. Click Update to regenerate the plot with your selected parameters. Click on Customize Graph to modify the appearance of the plot. For this plot, we can change the colors of the boxes. Click Update to apply your customizations. Scroll down to view the customized box plot. This graph plot the species richness for each of the different landforms. Species richness refers to the total number of species in a community or specific environment. Click Export to download your plot. Each unique plot should be saved using the Export button. As before, select the export format from a drop-down menu. We can select from PNG, TIFF, PDF, SVG, and PostScript files. Next, select your desired export resolution. Options range from 72 dpi to 600 dpi. Finally, select export size of either full page or half page. Click Submit to generate the export file. The download link will now update. Click it to open the export file in a new tab. Click on the Results banner to open a list of downloadable data files. Click on a file name to download. Continue to the Beta Diversity tab. Beta diversity measures the difference in species composition between sites. To start, select whether to input original data or normalized data. Next, select the number of permutations used to assess the Permanova hypothesis. This test assesses the relation between groupings and beta diversity. Following that, select a grouping variable for site or sample stratification. Finally, select a methodology for measuring dissimilarity distances for the Permanova test. Click Update to apply your changes. Click on Customize Graph to adjust the presentation of your plot. For this plot, we can adjust the color palette. Click Update to apply your customizations. Scroll down to view your customized box plot. The box plot visualizes the distance between samples and centroids based on groupings. For this example, the landforms are grouped along the x-axis, and the distances to centroids are measured along the y-axis. Click on Export to save your customized plot. After adjusting the export parameters, click Submit to update the download link. Click on the Results banner to open a list of data files available for download. Click on a file name to download that file. Click this link to view the detailed data table. In the New tab, we can view a table of statistical results for the grouping variable and residuals. Right-click the download link here to save this table. Once ready, continue to the next submodule rarefaction through the left-hand menu. Rarefaction is a statistical technique that assumes total abundance differences between taxa arise from sampling differences. This methodology assesses whether rare species have been sufficiently observed or recorded during sampling. First, select whether to input original data or normalized data. Next, select whether to calculate standard errors for estimating species richness. Finally, users can specify a subsample size for rarefying the community. If nothing is entered, the formula will default to the minimum sum of species counts across sites. Click on Update to apply your changes. Click on Customize Graph. Users can input a step size for sample sizes in the rarefaction curve. The line colors can also be adjusted. Click Update to apply your customizations. Scroll down to view the rarefaction curve. This graph plots the sample size against the total number of species. Continue to the Rarefaction Scatterplot tab. The Rarefaction Scatterplot displays the observed species count against the rarefied species count. Adjust the parameters as needed. For the scatter plot, users can also change the randomness parameters by adjusting the seed. Click Update to apply your changes. Continue to Customize Graph. For this plot, we can adjust the point colors. Scroll down to view the customized rarefaction scatter plot. Click Export to download. Under the Results banner, you can download the related data file. We will now proceed to the Species Abundance Distribution submodule. The Species Abundance Distribution submodule characterizes how species abundances are distributed within a sample or community. The Abundance Log Series Distribution tab fits Fisher's log series to abundance data. For this test, we can select the data input as normalized or original. Then input a sampling unit. Update to apply your changes. Click to customize your graph. Adjust the bar and line colors as needed. Scroll down to view your distribution plot. This plot shows abundance octaves against the number of species. Remember to export your plot. Download the related data files under the Results banner. Continue to the Abundance Log Normal Distribution tab. In this tab, we can use traditional binning to split species frequencies into doubling octave classes for Preston's log normal model, or go without binning to fit truncated Preston's log normal. Adjust the variables as needed. Select whether to apply the log normal trend line. You can also input a species abundance cutoff in log 2 units.
The default is -1.
Update to apply your changes. Click to customize your graph. Adjust the bar and trend line colors as needed. Scroll down to view your customized log normal plot. The trend lines represent binned and unbinned data results, as seen in the legend. Download data files related to this plot under the Results banner. Move on to the Ranked Abundance Distribution tab. Rank abundance distribution plots, logarithmic abundances in decreasing order or against ranks of species. Adjust the input data type and sampling unit parameters as needed. Click Update to apply your changes. Scroll down to view the updated plot. The abundance rank, or species abundance, is measured along the x-axis, while the relative log abundance is plotted along the y-axis. The most abundant species is rank 1. The slope of the trend line reflects species evenness. Remember to export your plot. Find the data files related to this plot under the Results banner. Continue to the Species Accumulation Model submodule. Species accumulation models study changes in species richness with increasing sampled sites. First, select whether to input original or normalized data. Next, select a species accumulation curve, SAC method. Options include exact, random, Coleman, collector, and rarefaction. Then you can select a nonlinear model to apply. Finally, we can select an interval calculation type. Update to apply your changes. Click to customize your graph. Adjust the line and box colors as needed. Select a confidence interval type, either polygon, line, or bar. Select the color for the confidence intervals. Click Update to apply your changes. Scroll down to view your customized plot. Species accumulation curves, SAC, show how the number of observed species increases as a function of sampling effort within a particular study area. Remember to export your plot. Download the data files related to this plot under the Results banner. Navigate to the Number of Unseen Species submodule through the left-hand menu. In the Abundance-Based Estimates tab, we can estimate the number of unseen species based on the observed species richness. Input the data type to use. Select the richness index on which to base our estimates. Input the number of permutations. If desired, select a grouping variable. Update to apply your changes. Adjust the box and border colors as needed. Update to apply your changes. Scroll down to view your species richness plot. The plot represents extrapolated richness index values by group. Click Export to save your plot. Download related data files under the Results banner. Continue to the Incidence-Based Estimates tab. Here we can again estimate the number of unseen species, but now based on species incidence-based estimators. Select whether to correct small samples. Select a richness index based on which to extrapolate the number of unseen species. You can optionally select a grouping variable. Click Update to apply your changes. Scroll down to view the extrapolated species richness plot. Click Export to save your plot. Download data files related to this plot under the Results banner. Finally, continue to the Functional Diversity submodule.
The functional diversity submodule requires trait data. Please ensure that trait data is provided.
Functional diversity FD explores how biodiversity influences ecosystem functioning, focusing on functional richness, FRIC, for the extent of the functional trait space occupied, functional evenness, FEV, for the uniformity of abundance distribution within the occupied trait space, and functional divergence, FDIV, for the extent to which abundance agrees toward the extremes of the filled trait space. Start by selecting one or more data analysis options. You can optionally select a correction method. Finally, select an ordinal method. Click Update to apply your changes. Download the related data files under the Results banner. Access all data files from this analysis session through the Download option in the left-hand menu. Click Results to expand the list of files for download. A list of all data files from this analysis session will open. Click on any file to download. The download.zip file contains all listed files. Download the WEGAN Docker file to recreate the WEGAN coding environment. Download the RSource file to get the exact R script used during your analysis session. After exporting your results, click Exit to return to the homepage. We hope you found this tutorial on the diversity module helpful.